Okay. Hello. Welcome to the stream. I am the world's best and foremost single cell based idol, Phi the Neutrophil, here after all the scuff. Holy cow. Everything was working fine up until 10:05 a.m. EST, and then my entire streaming setup went to absolute trash. Hello, I'm so glad to see you all here. Welcome, Engineer. Welcome, John the Eosinophil. Welcome, Variables. Welcome, Rogue. Welcome, Mirkala Cardstein. Hello. Welcome to all of you. Hello, Alice. Thank you for coming in. Did you enjoy the new loading screen? Yeah, so that's something that's happening. Hi, hello, welcome to the stream. Yeah, okay. Let me get a little set here. Today, I'm going to talk a little bit about rabies. But before I do, let's just chat a little bit. I want to give you all a little update as to what's been going on. So, just chatting, waiting for people to come in properly. Hello, Sign. Hello. Welcome. Yeah, so right now, my students are beginning to do their final projects, which is great. So what they're going to do is they're going to propose a novel idea in biological engineering, and it's the most fun in the entire world because we get to do all of the fun parts of science without all the hard parts of science, right? So getting to hear from students what their ideas are in terms of what they would want to make given the time and ability to is incredible because, I feel very lucky to be teaching this course because it represents a moment by which a student transforms from just someone who takes in information to someone who can actually output good ideas. And I always find that to be such an important moment in a person's academic career. And I just get to feel good. I don't have to worry too much about how their projects are going to work out, but getting to hear from their minds is such a rewarding experience. I have a question that I want to ask chat today. Because it's kind of related to this feeling of like, I don't have to worry about things not working out. I kind of want to ask chat, do you feel imposter syndrome at all? And this is not the topic of the stream today, but I kind of want to hear, put up a poll, because I've been thinking about imposter syndrome for a while. Let's do a poll. Because I'm genuinely curious as to the ratio of which people in my audience at least, feel this phenomenon called imposter syndrome. So if you don't know what imposter syndrome is, it's basically the feeling that you are not qualified to do whatever it is that you are actually qualified to do. And so this can take a lot of forms in terms of like self-doubt, self-sabotage, not believing in yourself. And I'm kind of curious because I'm weirdly somebody who I think I am very good at not feeling imposter syndrome because I feel like I'm pretty good at knowing whether or not I'm an imposter in that kind of way. And I'm kind of curious as to how everyone else feels that if they feel that way. Definitely, you do. Yeah, yeah. A lot of people do feel like they are not qualified for the things that they would like to do. And for me, I'm actually, I think I have a pretty good idea whether or not I am an imposter. So I think what I'm curious most about in this phenomenon is what that manifests, what it's like to have imposter syndrome. Because if someone tells me I'm doing a good job, I'm going to take them at their word for it. Luckily for me, I have no qualifications. That was my feeling. So I my chat knows this, but I spent most of my life being pre-med, and I felt unqualified to be a doctor. I felt like I wouldn't be a good doctor. And I had certain friends that would tell me, oh, it's just imposter syndrome. But after talking it out and realizing I would make a terrible doctor, it was actually an imposter that time. But I think that's true for me. It rings true. So I'm wondering what sort of, how that imposter syndrome manifests. Like, I don't understand, just the lived experience of being told that you're good at something and then not believing it, because I'm someone with a huge ego. I think I can do anything even though I can't. I don't know if it's imposter syndrome because I don't know if I'm qualified rather than feeling like I'm just not good enough. So I think that's the kind of nuance that gets left out a lot in discussions of imposter syndrome, because sometimes that's true. I'm not saying that that's true in your case, but sometimes it is true. And I'm wondering what factors what pieces of information you value when it comes to evaluating your own sense of self-efficacy. And I have a somewhat professional interest in this kind of thing because I am an instructor, so I do have students. And, students are amongst the more vulnerable to imposter syndrome. And so getting an insight from people as to how they weight information, I think is actually kind of interesting. I guess for me, I don't seek out the thing I'm good at, illustration, because I feel like I'm not that good, even though I know I have knowledge and skills in art and design. So I have a question for you, Alice. Do you still find illustration fun? Because that I think is kind of an interesting question. I tend to find reasons to doubt what they've said if it's positive and undue skepticism. I'm glad that's an interesting comment because you're aware of, you're taking a skeptical approach to your own skepticism, which I think is very interesting. It's just vibes? I guess that's fair. I have exams in a couple weeks. Am I qualified to do well with them? I'm not sure. Yeah, that's kind of hard to know. And I think being in the life stage of that's a good point. When you're still a student, you don't have cumulative life experience, and you can't because you're age gated. But not only that, because classes and your environment changes so frequently, right? You're only in classes for a semester, so there's no way that you can build up that kind of confidence, because after you're done with the class, if you did well, you're just going to take a harder class next time. So that's an interesting environmental concept with regards to imposter syndrome. It's pretty interesting. I think accomplishing a difficult task over praise would be the way to go. Accomplishing a task is more objective, but someone's views are subjective. I think that's definitely true. Although I told the story of my PhD advisor who successfully defended their thesis at an Ivy League college, still experiencing imposter syndrome. For some people, it's like rooted very, very deeply. And I'm really curious as to the mechanisms by which that happens. I love illustration as it does allow me to test my skills and really push myself. I feel so hard on myself for the failure. I have to balance it out by being sure to see success. I think that is kind of the tragic cycle of someone who does art. Although, like, I guess I'm wondering what initial factors are between you and I in terms of like why I am more able to free myself from that expectations. Not to say that I'm not hard on myself, but at the same time, like, yeah, I don't know. Because when I was illustrating, when I started, I totally sucked at it. And I was like, I would not make for a good artist. That's not imposter syndrome for me personally. It's just like, I would not make a good artist. I'm not having fun, and I suck at it. So I think those were the qualifying factors for me. But you actually still do have fun illustrating. For me, if I have fun doing something, there's no way I can be an imposter because I'm having fun. And I know that's not to say that that's advice, because I don't know how you internalize that. I'm just wondering why some people are having fun doing a thing, but still are that hard on themselves. I'm interested as to the intricacies as to what happens in the brain in that regard. I just have a difficult time taking praise while getting better at it. However, yeah, I don't know. I've never been, if people compliment me, I let it get straight to my head. I think it's really interesting. I figured it'd be a matter of high neuroticism. Interesting. Negative emotions more strongly. Yeah. That could be it too. Some initial factors. That does make sense. Intrinsic, extrinsic motivation. Oh, this is such a good conversation. I would love to have this conversation with a certain psychologist in the future, and this is giving me a starting point to think about what to say. But all right, thank you so much for that conversation. I think that is going to give my brain something to chew on for a while. But now let's get into talking about some rabies. Why don't we, huh? All right, let's get some lo-fi up. Let me know, chat, if the lo-fi music level, volume level needs to be adjusted at all. Okay, some announcements. So I have finished writing my Prion video script, and so because it's such a long and detailed and ambitious project, I'm expecting to take about a month to finish the video. There's something happening on 5/19, which I teased in my loading screen, and I will release a video about DIY biology and biohacking at some point. For those of you who tuned in to my Slay the Spire stream, you'll have known that that's what I've been working on recently. I'll probably release it either during video editing process where I'm inevitably going to burn out on this Prion video, so I might pivot to keep myself from going mad and releasing this biohacking thing, or I'll wait till I finish because I'm really excited to do this Prion video. How long will the stream roughly be? I think between an hour to an hour and a half. It's a pretty short presentation. Okay, so let's begin. So the paper that we're going to be talking about is called Toll-like receptor 3 plays a major role in the formation of rabies virus Negri bodies. Yeah, so if you didn't understand the words in this title, don't worry, we'll go through everything. So with journal clubs, I like to take a paper, an actual high-level science paper, and break it down for you figure by figure, just to give you, if you don't have a rigorous biology education, a peek behind the scenes as to what real scientists are reading and how real scientists tackle real problems. Because a lot of science media is simplified for an audience, which is fine, but I think you lose a lot of the excitement that comes with being a scientist if you only consume media that has been simplified for you. So I think it'll be kind of fun. Yeah, prions, yeah. Couldn't take it in high school, but trying to learn as much as I can now. Yeah, prions, very scary, but there will be hope. I don't like to fearmonger, and so even though rabies and prions, all these things that I do cover are very scary, but I hope that science will bring light to these terrible diseases and actually give us some hope for the future. That's very much my goal here. Okay, so we're going to be looking at real data, which is very exciting. So strap in and let's talk about some rabies stuff, all right? Okay, so rabies, list of virus 101. If you're watching me and you somehow haven't watched my rabies video, which is by far the most popular thing I've ever made, you should go watch it. It gives you a brief overview as to what makes the rabies virus so terrifying at a molecular level. So I don't like showing like gruesome images of like patients suffering. That's not what I'm here for. But it will tell you molecularly the strategies that the rabies virus takes that can be quite terrifying. So this is a double-stranded RNA virus. That's just the class that it belongs to. Rabies is a virus that infects neurons and is spread through saliva. It's almost always deadly, having a very, very high mortality rate. But luckily for humanity, vaccination is the most effective prophylactic treatment. So even though this virus is so insanely deadly, there is a highly, highly, highly effective treatment in the form of a vaccine, which is great. And if you really don't know anything about rabies, it's the inspiration for a lot of things like zombies and vampires, etc., because it is spread in rabid animals through their saliva, which means that if you get bitten by one, the infection can spread. So it is very, it evokes horror and the imagination of a lot of horror writers. So I'll come back and watch later. I'm revising polio mechanisms that hijack ribosomes at the moment. Interesting. I mean, polio is something that I might look forward to, look into the future to cover, because there's just so much to cover in biology, right? The thing about what I pick for topic coverage is that there are so many good ideas that I want to do as full-length videos that I only stream stuff that I'm not going to do as videos. So the fact that I've already done a rabies video means that I can stream about rabies. That's kind of my philosophy there. Okay, so rabies is an incredibly immuno-evasive virus. So you should know that your immune system fights pathogens all the time, bacteria, fungi, viruses, amoeba, whatever, you know. And so over evolutionary time, your immune system and these bad guys have evolved a kind of dance, a battle plan, if you will. And over time, this battle got more and more complex as one side tried to outdo the other in the long protracted battle known as evolution. And so immuno-evasion is a term that refers to the fact that pathogens have evolved to dodge the immune system. So rabies is incredibly immuno-evasive, and it evades the immune system in a lot of different ways. So it infects privileged tissue. This is a term that used to, it's kind of outdated by now, but basically because rabies infects neurons and because neurons are protected somewhat by the immune system, rabies gets away with a lot. So basically, neurons are post-mitotic, meaning that they don't replicate anymore, except in a few regions in the brain occasionally. The body has evolved to keep the immune system away from the nervous system because you don't want immune cells to be doing their work all willy-nilly in the immune system, right? If you have an immune reaction near a nerve tissue and you're not immune privileged, you might accidentally kill that neuron, which won't replace itself. Will this be uploaded as a replayable video? Yes, this will be archived. Rabies is also capable of inhibiting a signal called interferon. So if you've ever watched any medical show, interferon is an immune signaler that activates the immune system, helps fight back pathogens. Rabies is capable of stopping that signal from going through. Rabies also inhibits apoptosis in neurons, so it keeps neurons alive. So apoptosis, sorry, I should pronounce it correctly, apoptosis. Apoptosis in neurons can alert the immune system that there's something wrong, but rabies can actually prevent that from happening, paradoxically, right? Because you would think, oh, a virus that infects neurons kills neurons. That is true, but it actually will keep the neurons alive for long enough to reach the brain. And if it reaches the brain, essentially, your life is forfeit. I thought rabies is negative sense, not double-stranded. Oh, you're absolutely right. Sorry. You are absolutely right. Thank you so much, Fires UK. Yes. That is an error that I have. I was thinking about TLR3 binding double-stranded RNA viruses, which we'll get into in this stream, but thank you so much. Yes, I did make an error there. Rabies is negative-stranded, not double-stranded. My bad. So not only does rabies stop neurons from dying and therefore buying it time to reach the brain, rabies also induces apoptosis in T cells. So T cells are immune cells that actually will detect whether or not a cell is infected and then destroy that infected cell, preventing the spread of infection. So, yeah, rabies, incredibly immunoevasive. Hello, Surprise Pants, welcome. This paper deals with asking the question, what other systems of immunity might rabies be evading? Or at least that's the narrative that I'm putting on it, because actually the paper that I picked didn't do such a good job at telling the story. So I'm kind of telling the story for them. Okay, so that brings us to the paper title, Toll-like receptor 3. So this is the immune protein of interest that we'll be talking about today. So what are toll-like receptors? Oops. Toll-like receptors are immune surveillance proteins. So these proteins are proteins that monitor the condition of the cell, and if they detect a pathogen or a sign of a pathogen, they activate systems that turn on the immune system, right? So the history of this receptor, so these are toll-like because they are receptors that share a lot of properties with the toll gene in fruit flies. So a lot of genes that we know about have been studied first in fruit flies because they're such a convenient molecular model for us, genetic model for us. So that's why they're toll-like, named after the toll gene in fruit flies. These genes make proteins that bind pathogen-associated molecular patterns, or PAMPs. These PAMPs are a broad class of molecules that pathogens make. So these PAMPs can take the form of DNA, RNA, or proteins, just things that pathogens tend to make that our immune system can recognize. And so TLR3, toll-like receptor 3, is a specific type of toll-like receptor that is highly expressed in the neuron. It's also expressed elsewhere. It's not just neurons, but for the purposes of our story today, we'll talk about its role in neurons. So the question that we want to ask is, what role does this immune surveillance protein play in the disease pathophysiology of rabies? Is it a friend or foe, or both? And so down here, I just have a graph that shows kind of the overall structure of the interaction of TLR3 with the immune response, right? So we have infection, the presence of pathogen produces PAMPs, then you get TLR binding, you activate the immune system. Yep. So let's talk a little more about toll-like receptor 3 in particular. So TLR3 is expressed in neurons and in other cell types, and they detect double-stranded RNA. So even though rabies is single-stranded, when the RNA is replicated, when it is making copies of itself, temporarily that RNA is double-stranded, and so TLR3 detects rabies as it is multiplying. And so when TLR3 binds this double-stranded RNA, it starts the interferon 1 response, and so that just turns on the immune system. To simplify things, it's not very well characterized in neuronal cells, actually, because the neurons, as I mentioned earlier, are immune privileged. So it's actually kind of hard to study immune proteins in neurons because they just don't make too much of it. So the question that we're asking here is, does rabies affect TLR3? And I kind of want to poll the chat. So science is more fun, I think, when you take an active role in thinking through these problems. So I want to give chat a little bit of time as I take a sip of my tea to think about what you would do to answer this question. It's okay if you don't have a formal biology background. I just want to kind of engage chat a little bit and kind of farm out ideas. So how would you, on an abstract level, what's a type of question or type of experiment you could think to do to investigate whether or not rabies and this immune protein interact, and how they interact? And it's okay if you don't have like a super detailed question, super detailed answer. Abstract points are okay too. I'm gonna take a sip of my tea. Any ideas? I just realized we have 20 people watching now. That's amazing. Hello, hello. Welcome to the stream, everybody. Thank you for watching this little guy talk a little bit about rabies. For those of you who may have just tuned in, we're talking about the role or the effect that rabies has on this specific immune surveillance protein. Put them on a date? That's a pretty interesting idea, whether or not we can study the physical interactions between the rabies virus and the protein. Putting them on the date is not a terrible idea, actually. And we're actually going to see whether or not that actually happens later in the paper. It's pretty interesting. I don't think putting them in a boxing ring will work. I mean, if I stretch the definition of boxing ring, as we'll see later in the paper, that's kind of what they tried. I wonder if you could just have the virus particles and the protein in suspension together and see if they bind. Excellent idea. Spoiler alert, they don't actually bind directly. TLR3 doesn't actually, at least the authors of this paper could not get the TLR3 protein to bind directly to the virus nucleocapsid protein. Very interesting. Put them in a dish together and see how they interact outside a host, perhaps. Yeah, a lot of in vitro answers. Very, very cool. Sang, what was the question again? So we have this virus, rabies, and we're asking the question if it affects this protein of interest, TLR3. That's the question. These are all great ideas, and the authors are actually going to do something like that, or attempt to do something like that. That's actually not the approach that they took. That's not to say that chat is wrong, but that's not the angle that they want to start with. So they want to start with something a little more broad than, because what you're asking, chat, is a very targeted hypothesis. Do they bind? Before that, you might want to ask, do they change the levels in the cell? That's a little more of a agnostic question, actually. So the authors ask whether or not rabies infection affects how much TLR3, this protein that we're interested in, how much protein is actually made. Okay, so here we have some real actual data. This is a Western blot. And so what a Western blot tells you is how much protein is made. And so the darker the band is, or the thicker the band is, the more protein, I should say, speak correctly, the darker the band is, the more protein is being made. And so I'll point out, hopefully you can see my cursor. I think you can, yeah. We have the TLR3 protein, and the darker the band is, the more sample the more sample it contains. So after 24 hours, we're comparing this column with this column. So this is the non-infected neurons, and these are the rabies-infected neurons. And we see by eye that these bands are roughly the same. And in the quantification, the bar graphs, we also see that they're roughly the same. After 48 hours, you'll maybe see the rabies-infected neurons make a little more TLR3, but this difference is not significant. So if we go through, darker band, more protein, NI is not infected, RABV is rabies virus, TLR3 is the protein of interest, N and P just refer to different rabies proteins. This is actually a positive control to see whether or not the virus actually infected the neurons. So the fact that we see a band for N and P protein means that our rabies virus is actually here. And then CX is just a protein loading control. So don't worry too much about that. That's just saying that we loaded the same amount of protein in each well. So interestingly, this important immune surveillance protein that is important for the detection of viruses, it doesn't seem to have its levels changed by rabies at all. It doesn't seem to inhibit the production of this immune surveillance protein. Question, why does rabies target neurons? How does it know those cells are so vulnerable to infection? How does it relate to the proteins on the virus and the neurons which to infect need to link? That's a great question. So why is always a tricky question in terms of biology? Because the answer is almost always because evolution. But to take an attempt at answering this question, over evolutionary time, rabies just by happenstance infected neurons potentially because a surface protein on the rabies virus was able to bind a surface protein on the neurons. And neurons are actually not that easy to infect, because you imagine neurons are really deep into the body, right? It's not easy to infect neurons at all. You have to get through a lot of protective layers of body before you can get to a neuron. And through those protective layers, your immune system is working. So one of the only ways that that could happen is direct exposure. And so it's this weird combination of the fact that because rabies can spread in the saliva, it can get access to the neurons. And all of those factors play into this big evolutionary story where it's evolved to be in this niche. So, interestingly, rabies does not affect the production of this protein, but what other ways could a virus mess with a protein? What else would you check? And chat, you're on top of it, right? All this idea of boxing rings and putting them together on a date. You might think to test whether or not rabies can actually bind to the protein of interest, whether or not it's doing something to it directly. But that's actually not where the authors go next. Not only can you test whether or not the proteins increase or decrease in levels, maybe rabies is moving the protein somewhere else where it's not as useful. So that's the next question that the authors ask here. So where does TLR3, this immune protein that we're interested in, where does this protein normally live? So TLR3 has what's known as a canonical localization. All that basically means is that in a healthy cell, TLR3 tends to live in a compartment called the endosome. So endosomes are basically vesicles, or you can think of them as bubbles that live inside the cells that are important for molecular delivery. So if you think about like Uber Eats, you can think of the car as a type of vesicle, a bubble that you can pack things inside and move inside the cell. So TLR3 lives in endosomes of other cell types, but because neurons are so understudied with regards to immunology, we don't actually know if that's true for the neuron. So here we're going to get to a piece of data, a piece of image data, which I think is so beautiful because I just like looking at fluorescence. This is great. So the authors did what's called immunofluorescence on neurons infected or not infected with rabies. So in this column, we have normal cells, and they used antibodies that are conjugated to fluorescent molecules to color the cells. And here's what they found. In green, we have TLR3, that's our protein of interest. In blue, we have the neuron nucleus. And in red, we have a marker called CD63, which is a protein that is found in these Uber Eats vesicles. And we get this very pretty picture here, right? And I can actually walk through what we're actually seeing here, because I think this is the kind of stuff that if you just consume science media, like from newspapers, you don't actually get to fully appreciate. Let's see. Let's make this bigger just so that we can look at these structures a little better. So we have the nucleus and we have these pockets. And what we're seeing is that when you combine green and red, that you get this overlap and it forms yellow. So what this tells us is that the TLR3 is co-localized with the endosome. If the TLR3 was not co-localized with the red marker, we would not see this yellow. So this yellow basically is an overlap of red and green, telling us that these two markers are very close to one another, which is very interesting. What if rabies hides by only having a low presence in neurons spreading out over a large number, which then creates this effect? Hence why this TLR3 thing doesn't get produced. So we're actually going to see the extent by which rabies can multiply in a neuron, and it's something that you might get creeped out by. So it's an interesting hypothesis, and we'll see whether or not that holds out further down into the paper. Okay, so we see green, we see red. When you overlap the images, we see yellow. That tells us that TLR3 is in the endosomes of nuclei. Very, very cool. What about when rabies is a part of the question? What happens to TLR3 when rabies is introduced? I should have just kept this big, actually, because I'm throwing up the next image. All right, rabies. What happens when rabies is introduced? And how does that compare to the canonical localization? So again, we have green for TLR3, the immune protein of interest, and we have red for endosomes. And let's actually have a look here. Instead of yellow, I mean, we still do get yellow, but instead of yellow, we have these green-only spots. That's crazy. Green-only spots. What that tells me is that even though TLR3 is it's almost completely co-localized with the endosomes, these pockets, these bubbles, rabies drags them out of those bubbles and puts them into these little things that are near the nucleus. What these things are isn't exactly clear. That's why the authors call them perinuclear aggregates. This is just a general term to describe the fact that rabies has somehow removed TLR3 from their normal location. This is actually quite important because when endosomes are formed, endosomes are compartments that traffic stuff from the outside of the cell inside the cell. So that's why TLR3 is in the endosome, because it's detecting whether or not a virus has entered the cell. And rabies is actually removing the detection protein from the place that it needs to be. Super, super weird, super, super interesting. Yeah, yoink, exactly. Yeah, rabies just yoinked that protein right out of that bubble, and at this point, it's unknown as to why or how that happens. So I think that's pretty incredible. So where did TLR3 go? So this protein of interest, this immune surveillance protein, no longer co-localizes with the delivery bubbles, and I've blown up this image here so it's a little more clear. Located outside the nucleus during rabies infection. What's happening? And why does rabies do this? The authors then go on to say that this doesn't happen with herpes simplex virus 1 or Sendai virus. So this is a property that is unique to the rabies virus because these other viruses don't seem to do that. Mechanism would be interesting to look at, but this is a paper from 2009, so more information may have come out, but I wanted to take a look at this because I just think this is such a beautiful paper. So rabies is putting TLR3 in the rubbish bin. That's actually not true either. You would think that rabies would be interested in just destroying that protein, but it's actually doing something more bizarre with the protein. Is TLR3 still active in these? We don't know. As of this paper, whether or not it's been found out later, we don't know right now. Okay. Interesting questions, chat. Interesting questions. So what would you check next? So it doesn't increase or decrease the levels of this protein. It just moves it out of the part that it's supposed to be in. So what else? What else? Is it going to misfold TLR3? Getting closer, perhaps. That's an interesting hypothesis, too. What would we check next? What would we check next? Interesting thing to think about. Well, what did the authors try? The authors wanted to know, and this gets back to the date and the boxing match stories. The authors wanted to know whether or not the proteins bound to one another. And so you can kind of approximate this by doing more of the same experiment, right? Instead of looking at the location of endosomes, does rabies virus overlap with the TLR3 marker? So this is the same sort of experiment. And so TLR3, that's in red here. NC is the rabies virus package, the nucleocapsid, NC for green. And we actually see that when we overlap TLR3, the immune protein, and NC, we get a quite nice overlap. So it looks like the rabies coat binds TLR3. So it's touching TLR3 somehow, or sorry, it doesn't bind, it co-localizes. They're located in the same area. The mortality rate of rabies, very, very close to 100%, 99.999, etc., etc. Can we replicate this effect as to see what the TLR3 behaves and is different to normal? Yeah, we could test the TLR3 function, and actually that's something that I'll talk a little bit about later. So we can test whether or not interferon has been affected. But here's the lovely thing about how complicated science can get. That's a correlation. So if TLR3 function is screwed up after you use the rabies virus, we don't know if rabies is doing some other thing to screw up interferon production, or whether or not it's this TLR3 getting sucked out of the endosome that's doing that. But that's an interesting hypothesis. Is it not removing it because it's trying not to mess with any mechanisms, sensing for a lack of TLR3? That's a very, very interesting hypothesis. I've not thought about that, but that's how other other systems work, specifically NK cells. So NK cells, natural killer cells, these cells are really good at detecting the lack of a immune protein on the surface of cells, because some viruses can pull immune proteins off of the surface. Some cancers actually pull immune proteins off the surface, and NK cells can do that very thing. So it's a fantastic hypothesis to have. Very, very cool. Very, very cool thoughts going on in chat. All right. So the rabies package seems to be overlapping with the TLR3, and the authors wanted to know spatially what that was looking like. So they wanted to get in and really see how the rabies virus is interacting with this immune protein. And so they do an experiment called confocal microscopy. So confocal microscopy is using a microscope to take optical sections. So what's an optical section? An optical section is taking an image one slice at a time. So I have up here a cartoon explaining confocal microscopy. So imagine you have a sphere. Confocal microscopy lets you step through the layers of that sphere and generate a 2D image. So, on the left here, we have the sphere in question. And as we take images, we can then get deeper and deeper into that sphere to kind of figure out what this thing looks like on a structural level. So we know that they overlap, but how do they overlap? We're not entirely sure. CT scan but for microscope. Yeah. Very true, very true. So how do the rabies virus and the TLR3 immune protein interact? Here's the crazy part. This blew my mind. This is so cool. When you take optical sections here, you actually find that the rabies nucleocapsid captures TLR3 into its core. So here's how we know that that's true. Look at here. This is the rabies nucleocapsid, and see here, there's a ring, a very subtle ring going on here. And on that same slice, the TLR3 is completely solid. So imagine a ring, and you can see it here in this image, a green ring surrounding a red ball. And as you step through that ball, what you get is this, a coat of nuclear capsid protein from the rabies virus covering the TLR3 protein dot. The rabies virus swallows the immune protein. What? That's crazy. I've never seen a virus do this before. Why? Why in the heck? I don't remember who it was in chat before, but someone suggested that rabies could be destroying the TLR3 protein by throwing it into a garbage bin. The cell does have stuff like that, right? It's called the proteasome. It's basically a garbage disposal for proteins, but it doesn't destroy the protein. We know that the protein levels don't change. What is it doing instead? It's capturing that TLR3 protein, putting it into a core. That's insane. That's insane. Is the virus acting like an immune cell? Depends on what you mean by that. That's really, really just incredible. Incredible molecular mechanism. So cool. And I'm dying to know why the rabies virus does this. So a very natural question to ask is, what do these aggregates do? do these aggregates have a function? Why does rabies steal this protein and like swallow it up? So there's this thing called a Negri body that has been observed with rabies infection before. And a Negri body is proposed to be something called a viral factory. So viruses, not all viruses, but some viruses, form a structure within the cell, an aggregate in the cell that helps them, and they assemble this aggregate in a way that it becomes a factory to pump out more viruses. So are these Negri bodies, these structures that this nucleocapsid forms around, are they viral factories? How do we figure that out? Well, what we can do is we can test for the presence of rabies RNA. So if rabies RNA is in these perinuclear aggregates, we could make the argument that these are factories. And what we see here, it's kind of hard to see, this isn't a great image, but we see that when you have a rabies infected cell, this aggregate lights up, which means we've successfully detected rabies RNA, meaning that these bundles have rabies RNA. So not only do they have the nucleocapsid and the immune protein, they also have rabies RNA, which means this is a rabies viral factory. It captures this immune protein and it's crazy. It's in a rabies viral factory. And why? Why did this happen? What possible mechanism could the rabies virus have used, or yeah, I'm getting a little ahead of myself. Why did this thing evolve this way? It's kind of nuts. So let's get into why this is happening. So this is the post that I teased in my community post. This is the rabies viral factory called the Negri body. There are other viral factories in other viruses that rearrange the cellular guts to power the Negri body. And we see here very conveniently that there are mitochondria on either end of the Negri body because it's a very energy intensive process to be making copies of the rabies virus, right? So the fact that it's like nudged itself and it's got access to these mitochondria and access to the endoplasmic reticulum. This Negri body is huge. Look at the nucleus. It's enormous. So it's pumping out these rabies viruses, all while keeping the neuron alive, because you don't want the neuron to die too early, because then you won't be able to make copies of yourself. Just nuts. Just absolutely nuts. Okay. So it captures TLR3, the immune protein, in this Negri body. The question that everybody wants to know is, do the viral factories need TLR3? Because that's an interesting question. Does it have to have that TLR3 in order to function? So how would you test this, chat, as I take another sip of my tea? And I'll go back and look at chat. How would you test the hypothesis that the rabies viral factories need TLR3? And I'll answer some questions from chat. How long does rabies take to kill someone? It depends on where the person is bit. The closer they are bit to the brain, the faster it takes. Could be anywhere from a few days to a few weeks. Rogue answered that. That's correct. Isn't this the way that we got to multicellular life? Yeah, so for those of you who watched my archaea video, the history of eukaryotes, as we believe it to be right now, is that an archaea swallowed a bacteria and potentially a virus, depending on who you talk to, and life just kind of went on from there. I would say it's not quite that way, because the host of this situation is definitely not benefiting from having consumed this virus. But it's like picking up junk and making guns with it. Yeah, absolutely. It's so creepy. Mitochondria is powerhouse of the cell. Yes. The question about rabies needing TLR3 is a cool question. Yes. Test it by preventing access to TLR3 in a neuron. Yes, that's actually the approach that the authors take. Prevent rabies from getting access to the TLR3 protein and see if it can still make viral factories. I was just about to ask, why is TLR3 needed anyway? So TLR3 is an immune surveillance protein. Generally, when we make TLR3, we make it because it's good at detecting RNA viruses. That's why we generally make it. But why does the rabies virus need it is an interesting question. Take TLR3 out or find something that binds TLR3 to deactivate it. Exactly. Yes, yes. Chat. You're all scientists. Yes, that's exactly what the authors did next. They removed TLR3 from the system. So they use a system called microRNAs that can seek and destroy TLR3 mRNA. So microRNAs are RNAs that bind to their complement, and when they bind, they can recruit an enzyme that destroys RNA. So if you destroy the TLR3 mRNA, you also destroy the protein. Yeah. So when you destroy TLR3, you can ask, can rabies still make the factories? So here we have an experiment where we have a negative control miRNA, microRNA, and we see that you make 100% of your TLR3 mRNA, not surprising. When you add that microRNA, you destroy that TLR3 mRNA, right? This makes sense. When you do this to a rabies-infected neuron, you find that the number of rabies mRNAs plummets. It plummets. It goes all the way down. See this? Look. Let me blow this up to show you how crazy this is. Let's get my OBS. Make the graph a little bigger. Look at this. Look at this. Transcripts. Rabies virus. Relative fold. When you don't destroy TLR3, you get 100% of the rabies mRNA being made. When you destroy TLR3, look at how low, look at how few rabies viruses are being made. So we can conclude from this that the rabies virus needs the immune protein to replicate. What the heck? That's crazy. That's actually freaking bonkers. I've never seen something quite like that before. That's truly nuts. That's a treatment, right? Ooh, we're going to talk about treatments at the end of the presentation, but keep that thought in mind. Maybe it's trying to prevent being found out by the TLR3 and it's needed more than two birds with one stone kind of thing. Exactly. So that's a hypothesis that the authors have. So maybe rabies is hiding the TLR3 so that they don't get found out by the immune system. It's definitely a good hypothesis. So this is a confirmation to show that destroying TLR3 destroys the rabies virus as well. So here we have an image. In green, we have a cell marker, GFP, green fluorescent protein. This just marks the shape of the neurons. And TLR3 in red. So in the normal non-infected cell, we have a lot of TLR3. And when you have a normal amount of TLR3, you also get a ton of rabies virus. And you see in the merge, it's kind of hard to see, but you see this magenta. This is happy rabies virus. It's doing its thing. It's stealing TLR3. It's making a ton of viruses. Okay. What happens when you destroy TLR3? What happens to the rabies virus? You destroy the TLR3, you destroy the rabies virus. Isn't that incredible? Look at how different. Look at how different this staining is. Look at how much TLR3, how much rabies virus is up here. And when you destroy that immune protein, you also destroy a ton of that rabies virus. Yeah, that's nuts. Okay, but that's not enough for scientists, right? On what, like this is pretty good already. This is an incredible finding, incredibly interesting. But if we're talking about evolution, then we have to determine whether or not this affects fitness, whether or not this makes evolutionary sense. So what's a next step experiment that you would do here, chat? Let's take some time to think while I hydrate. How do we know that this actually matters? Like, this might just work in cells on a dish, but how do you know this is real? How do we know, chat? What's some convincing evidence to prove that this is a real result, that this matters for actual living people? Because all of these experiments were done in cell culture, meaning it's just cells on a dish. Gotta do in vivo studies on something and test some mice. in vitro studies. I think you probably meant in vivo. Yes, in vivo. In vivo, for those of you who don't know, means in a live animal of some kind. So that's exactly what the authors did. So good job, chat. Let's do it in an animal. So here's a warning. It's always tricky to talk about animal research to the public, but the next slide will detail some research in an animal model. So if you are not a fan of that description, tune out for a little bit. But let's move to a mouse for the rest of us. Yeah, exactly. PlayStation, exactly. A mouse. All right. Send in the genetics. Yeah, exactly. So both of those answers are correct. We're going to do this in a mouse, and we're going to use the power of genetics. We're going to use a knockout mouse. So these are special mice that have had their TLR3 gene destroyed. So these are mice that no longer make TLR3. And when you give these mice a rabies infection, we actually find that the mice that don't have the TLR3 survive longer. So these are days post-infection. We can see here the wild type, meaning the normal mouse, when they are given, when they are infected with rabies, half of them die by, let's say, day 13. And then when you knock it out, they survive a bit longer. This data is not that interesting to me because the difference here is not that striking. They could have probably gone for longer because this data, it's just not that different, but I do buy that they survive a bit longer, although I would have liked to see a bit more time. What's striking here is checking how much rabies virus is being made in these animals. And indeed, when the mice completely lack this immune surveillance protein, they make a fraction of how much rabies virus is in the wild type. They still die. You know, it's not perfect. But it does seem to really actually matter, which is just nuts, right? Somehow rabies is using this TLR3 protein in the viral factory. It's a key component to making more virus. So there's more stuff in the paper, but that's the story that I wanted to tell. So here are some conclusions to say. The rabies virus actually needs our immune surveillance protein TLR3 to make these viral factories, also called Negri bodies. The TLR3 is packed up into the core of the viral factories, so we know how it interacts. Mice that don't have TLR3 survive rabies infection better, so that provides good evolutionary evidence to show that rabies evolved to need this as a part of its reproductive cycle. But isn't TLR3 important to overall immunity? That's definitely true, but it's about trade-offs, right? So TLR3 is important for rabies infection, and maybe that's why the virus why they're not immune to rabies infection, right? Maybe it's like you remove an immune sensor, which is bad, but it's also good because rabies can't use it as well to make copies of itself. They are important, and it's a trade-off. That's why these evolutionary battles are so interesting and so complicated sometimes. That's a good thought. So yeah, so I'm dying to know why or how the rabies virus is using the TLR3 protein. I want to know the molecular mechanism. Like, what is it actually doing? So let's talk speculation. So here's where I don't know if more research has been done because this paper's from 2009, so it's been a while. But hypothesis one, as a lot of chatters have correctly or have reasonably suggested, by removing the TLR3 protein from its normal location, rabies has once again blunted the immune system. So that is one hypothesis. We know that rabies decreases interferon signaling, so that's one piece of the puzzle, and we know that rabies decreases TLR3 in the correct location. We know that TLR3 leads to interferon signaling, but these are correlates, right? These are correlation, they're not causation. So any scientist worth their salt might be interested in testing whether or not removing this protein is the thing that decreases interferon signaling. Is it possible that there's another factor by which it's not 100% survival still? Yeah. So just because rabies needs it to make the factory doesn't mean that it can't do it. Like, it needs to make the factory well, but it can make kind of crappy factories and still get away with it. this is kind of a hard hypothesis to test, right? Because these are correlated, it's kind of hard to rule out all possible other explanations. Maybe you could break the rabies virus so that it can't remove TLR3. And if it can't remove TLR3, we can check the interferon. I think this is a reasonable strategy, but I don't know if there's a broken rabies virus that stops TLR3 aggregation. This is the beauty of science, right? Like the beauty of molecular biology is being able to tease apart correlation from causation. Give them pseudo TLR3 and see if it still works. That's interesting. What properties would that pseudo TLR3 have to have for that to make sense? I don't quite understand what you're suggesting. Exonisis. You could add back TLR3 to infected neurons, but again, because TLR3 is correlated with interferon or is causative interferon signaling, you wouldn't really be able to tell that apart. Correct location. What about putting TLR3 back to where it should be? Yeah, that would be a great approach. I don't know how you would do that. I added here, break up the factories and see if interferon is restored. So that is actually not as good as, Alice, what you were saying. If you could just put TLR3 back into the correct location without breaking up the factories, that would be even cleaner. Great suggestions from chat. Fantastic. Oh, I love this so much. I love that you all have such good ideas. And it's really given me proof, because I don't know how many of you have rigorous biology education, but it's given me proof that everybody can understand science, right? Everybody has the brain for this. Maybe not to do science professionally, but everybody can engage with high-level science. You just have to care enough. So it's been so much fun. Okay, so some other speculation. Maybe by removing TLR3, rabies cuts off a way for the cell to commit apoptosis. So I wasn't quite forthright with chat. TLR3, it does the interferon signaling, it activates the immune system, but you can also cause the cell to blow up if things are that bad in the cell. TLR3 activation could lead to apoptosis, which denies the virus a place to replicate. If you're a fan of Dota, you know all about denial. Citizen science, yeah, exactly. Didn't even take high school biology. Yeah, exactly. I'm so proud of you, Alice, because there are people who self-sabotage in my class, and I teach people who are very dedicated in learning biology. And the fact that you're not afraid to just go out and say something, and the fact that you're on the nose a lot of the time means that, I think you will go far in terms of learning. That's a great attitude to have. Okay. This is also hard to test, right? It's got all the same problems as hypothesis 1. Breaking up the factories. Again, I don't even like this wording anymore, because truly just removing TLR3 is the way to go, because breaking up the factories introduces more confounding variables. But I think these are both interesting hypotheses. They're not really mine, they're the authors, but they're good. Welcome, Thomas, the thermonuclear bomb. Hope your exams have been going well. We're actually at the end, unfortunately, of this crazy story, but I highly recommend. I think this VOD is pretty fire. This was such a fun stream. Let's see. So, caveats. I actually have enough background in this kind of research to actually know where the limitations might be. So a lot of this research was done in cell lines instead of primary neurons. Ultimately, though, because they ended with a mouse, a lot of those caveats might not be that important. So I can't really think of other criticisms I would make. I'm just so blown away by how amazing that finding, that the viral factory that the rabies virus makes captures the immune system protein. It's just absolutely nuts. Software engineer, but pharmacology was my second choice. Fire Ice, you're actually very knowledgeable about my field. I could have sworn that you could have been a professional. That's awesome. Is rabies virus located in the brain or other neural cells in the body? So it starts in your motor neurons, heads up towards the brain. Could you destroy the cells even though it created issues like paralysis? If it saved the person in any way, part of leg. Yeah, so T cells So there are rabies viruses that are not very good at stopping apoptosis. And when they can't stop apoptosis, the T cells can actually deal with them a little better. And if rabies has their ability to stop apoptosis removed, they actually can't infect very well. I wonder if other viruses might do something similar, or if it's just rabies' unique thing. So we know that HSV, herpes simplex virus, and Sendai virus don't do it. Whether or not other viruses do it, I don't actually know. So I probably should not eat TLR3 if I have a rabies infection. Yeah, that's true. You like TLR3? Too bad. Just make sure that you don't increase your levels after... Well, actually, we don't know that. We don't know if more TLR3 would change the rabies virus. It makes sense, but we just don't know that. So I took a quick look into research since then. And so there's this paper in 2016 that used fake double-stranded RNA to distract the TLR3 protein and stop it from being assembled into factories. We've actually seen that this helps the rabies virus work. So it's not direct evidence. So this is kind of getting into maybe what Alice was hypothesizing. Or not just Alice, but a couple of you in chat were hypothesizing that removing the TLR3 from these factories is a good therapeutic strategy. So by using this decoy RNA, you can stop the TLR3 from getting into those factories, and that actually helps the vaccine work even better. That's pretty interesting. Another paper that I found was inhibition of TLR3, so stopping TLR3 function, severely decreased rabies severity in mice. That's a more recent paper in 2022. So it's a little different than a knockout, but this idea that TLR3, like lowering, like anti-inflammatory targeting at this level, does seem to be a valid therapeutic strategy, although I don't know if this has moved into clinical trials at all. And yeah, that's all I had for today. I thought this was super fun. I love this paper. Fun story. I actually read this paper before and I totally forgot. When I was in graduate school, taking classes in my first year of graduate school, we had to do a novel research project presentation, a proposal basically. So as scientists, we have to practice generating new ideas. And the thing that I want to do, because I've been obsessed with rabies for quite some time, you know, I'm a hag now, but back when I was young and spry and in grad school, I wanted to do a rabies proposal presentation, and I actually read this paper for that article, for that project, which was very interesting. I want to address something that I didn't really quite cover. For all of you wondering whether or not TLR3 and rabies directly bind, they don't actually seem to directly touch one another, or at least it's hard to prove that they do, because the authors did what's known as a pull-down assay, or a co-IP, co-immunoprecipitation, for those of you who want to Google it. That's an assay that basically tells you whether or not two proteins bind inside of a cell, and they could not get those two proteins to bind. So TLR3 and the rabies nucleocapsid, there's empty space in that viral factory, not empty space, but there's other stuff in that viral factory that might connect the two, but they don't seem to bind very strongly, if at all, which is even weirder, right? Like it's capturing the TLR3 protein, but it doesn't actually need to touch it. Just bizarre. Next, try TLR3 inhibition plus induced coma. I have mixed feelings about the coma part of the Milwaukee protocol. If you've watched my 5-bit, you know all about that. This is mind-blowing. Definitely wasn't expecting this. Yeah. Molecular biology. Super interesting. My favorite stuff to cover. Do you think Foldit is a good place to start learning microbiology, or should I start from some book or something? I don't actually know what Foldit is. I'm not a biochemist or a biophysicist, so I'm guessing Foldit is a protein folding thing. I would just start with like Khan Academy or my YouTube channel, selfishly. If you want to get into molecular biology casually, Stuff like SciShow, Khan Academy, that's a good place to start. There's no replacement for school, though, unfortunately. Like, there are good free resources. You can even take classes at community college for much cheaper. I do plan on doing like a AP Bio thing. I was going to do it this semester, but my job is so busy during the school year that I might like do it over the summer actually to help AP biology students. And that AP biology is a great place to start like learning the cool things about molecular biology. I really, really want to see someone do a study with modified TLR3, adjusting its structure but not its function. And really want to see how TLR3 differs in opossums if they can survive it. Yeah, that would be really interesting, right? Because opossums are like rabies resistant. Yeah, that would be interesting and not something that I considered. So that's very cool. Kind of like a chemical bond, like a smaller version of how the ovaries and fallopian tubes. It's like a it's kind of depends on what you mean by a chemical bond, right? So they're not covalently linked. So covalent bonds are like the bonds in between molecules that share electrons. So the rabies virus and this TLR3 protein, they interact, but we don't really know what the nature of that interaction is. Is there a missing link, like on how rabies separates TLR3? We don't know. Presumably that's true, because how else would that happen? But we don't know if and how many, if there is, and if there are linkers in between the rabies virus and the TLR3 protein. It's interesting, very interesting. I only did medicine because I like biology. A lot of people think there's no critical thinking to it. Oh, interesting. I'm kind of wondering where you are at in your medical career. Critical thinking in biology. I mean, when you learn it for medicine, there's not really critical thinking. Like, you just have to know the biology so that you can do the critical thinking in the clinical setting. But biology research is all about just the basic building blocks of the scientific method. It's excellent just to learn how to think, which I can't say for other disciplines. What's the difference between molecular biology and biochemistry? So molecular biology is a broad field. Biochemistry is under the umbrella of molecular biology. Molecular biology can include genetics and cell biology stuff. Well, cell and molecular are different, but molecular biology can include genetics, for example. I will probably go back to school for biochemistry next time I get fired from my tech job. So sorry that you have to deal with tech layoff like constantly over your head. Certainly don't envy people in that position. If you are thinking of transitioning from tech, you know, AI is getting big in biology. That might be something to hop onto. It could be interesting. I wondered if maybe the TLR3 was being digested to make more RNA or amino acids for more viruses. So we actually know that the TLR3 levels don't change, but that doesn't actually stop your hypothesis from being right. We know that the level doesn't change, but whether or not the rabies virus is destroying it and the cell is making more of it to compensate, we don't know yet. Or I don't know as of this paper. That's a very interesting hypothesis. Make more of these, Fi. Love it. I really want to. This was so much fun. I think this is maybe one of the best streams that I've had in terms of talking about stuff that I like. It's just so unfortunate because I don't want to cover things before I make a video on them, right? Like, I don't want to do a Prion stream until I make my Prion video, because as much as I love streaming as a hobby, my bread is buttered with the long form video, right? Like, I actually make money on those. It's really fun, and a lot of people watch them. So I'm like saving my best ideas for videos, and then after I do the videos, I'll do them as streams. It's very fun. Almost finishing your first year of medical school. I'm guessing that's medical school. Congrats. Honestly, it's tough, the first year of medical school. A lot of stuff that you have to learn. Yeah. This was a lot of fun. Yeah, like the moment that I'm done with that Prion video, sometime in June, I will do like a Prion stream. What else might I like? So I'll take suggestions, not of pathogens, because I want to cover pathogens in long form, because I think the interaction between viruses, bacteria, fungi, and the immune system is the perfect biology story because there's so many back and forths. And I think that's part and parcel of a good story. So Prion video comes next. I might do biohacking before that if I burn out editing the Prion video. I wrote the script. It's like 12 pages long. It's insane. So it's going to take me a very long time to edit it to the quality that I want it. But in the meantime, like I would love to do like similar things, but like for the mitochondria, or maybe just another rabies paper, or tuberculosis, or gonorrhea, right? So I'll put a poll up actually. So I want to know, because again, I'm saving my best ideas for videos. Could I do something autoimmune? So my stance on autoimmunity is it's almost too tragic for me to cover as a long form video. I might do it as a journal club though. So I'll actually add that because, you know, I'm a little cartoon guy and I make jokes in videos and I don't feel like doing autoimmunity as a joke. My videos aren't jokes, but I don't want it to be conceived that I'm making light of autoimmune disorders. So let me do a poll. So yeah, there's probably other rabies stuff that's interesting. Tuberculosis is another good option. I thought about doing tardigrade, but I'm kind of tapped out on tardigrade stuff. I think I covered what I wanted to cover for a while. Gonorrhea is a good option. What else did I do? Rabies, tuberculosis, gonorrhea, archaea. I could do an archaea journal club. That could be fun. Actually, I'll leave this one to be autoimmune because y'all seem to be interested in that, and I don't mind doing that one as a stream. Okay, pulls up. Welcome, female Thunderbirds. There's an achievement in Play Game called Not Another Zombie Game where you don't create a single zombie and kill everyone with a single strain of rat rabies. Interesting. If you want to make something on tuberculosis, you could collaborate with Hank Green, who's decently big. Oh, yeah. Just casually collab with freaking Hank Green. I applied to work for them and I got turned down, unfortunately. They were hiring for the tuberculosis project, and unfortunately, they just didn't hire me. Yeah, which was a little unfortunate, but I'm happy because it kind of forced me to work on my own stuff. Yeah, that's a fun fact. I applied to work on that tuberculosis project and got rejected. For autoimmune, there are some less serious conditions like Hashimoto's. I have that personally and it's pretty common. Interesting. Yeah, I don't want to cover something that's just gonna like bum people out. Not to say that Hashimoto's doesn't bum you out occasionally, but you know, it's a delicate balance because with things that cause human misery, you don't ever want to be too like laissez-faire with it. Neutrophil doesn't want to do autoimmune. Yeah, I mean, it would be bad for my brand if I did autoimmune, right? We don't want to be a bad guy. That's their bad. They're missing out. True. Tetanus. Yeah, again, if I cover tetanus, it would be as an actual video instead of a stream, because I always want to do the best ideas as videos first. DHT hair follicle biochem. Don't know what you mean by that. Although there's all sorts of autoimmunity that can happen at the hair follicle, as I learned from Cells That Work Black, interestingly enough. Cystic fibrosis. Hate how Hank hated on corsets in it, though. I don't actually know what that means. I would have to read a lot more into that to be comfortable talking about that kind of thing. Cystic fibrosis, that would be under the umbrella of molecular biology. That's, correct me if I'm wrong. I'm going off the cuff. You can totally, like, roast me in chat if I'm wrong. But that's a point mutation that causes a disease in the lungs? Or am I getting that mixed up with something else? So it looks like overwhelmingly we are in favor of something to do with autoimmunity. So yeah, I'll try to find an open access autoimmune paper for the next stream whenever I get to it, because I'm pretty busy in May, actually. Once I hit June, things will be a lot less busy in Find the Neutrophil land. All right. Okay. I can understand why you are cautious about doing an autoimmune video, especially with the HIV epidemic of the 80s and 90s. Yeah, that's the thing, right? Like, HIV is the poster child of autoimmune disorders. Not autoimmune, sorry. is the poster child for like not quite autoimmune, but HIV. Sorry, so I guess these are two unrelated ideas in my brain. HIV is like such a good like virus to cover in an autoimmune context, but again, like gonorrhea, it's not that serious if you get it treated. There is treatment. Rabies, there's a vaccine, so like you're probably good as long as you can make it to a hospital. HIV for a lot of people is still a big freaking deal, even though there are very good medications out there, but not everybody has access to those. And you know, it can just like make your life a lot harder, and it's not something that I'm very comfortable with covering. Cystic fibrosis affects the fluid in the body, making it thicker. Yeah. I think I learned it in the context of the lungs. Allergies are lighthearted for the most part, but they're also not interesting. I don't know if that's necessarily true. Allergies might be the best thing for me to cover, actually, because it's variable and it can be quite severe. Like a deadly peanut allergy could be interesting in terms of understanding why some things like dust are usually not a big deal, but something like peanuts are. That could be interesting to look into, although I don't really know if there's a story there. Although if you finished your first year of med school, maybe you know better than me whether or not there's something interesting in there. All right. Well, that's probably going to be it for me. I had so much fun today on stream. This is a paper, since I've read it before, I was very comfortable with talking about it. Oh, I want to do one more poll. Yeah. Wait, don't leave yet. I want to do one more poll. I want to do one more for those of you who stuck and enjoyed the stream. You know, if you enjoyed the stream, you know, I'm being a little presumptuous here, but if you enjoyed the stream, I want to know your level of biology education. I'll put that up as a poll because I want to know if I'm doing a good job of catering to a wide audience. Making that poll right now. Yeah, tell me your we'll say required class. Biology undergrad. And then post-grad professional. I kind of want to know who's in the audience, so I continue to refine how I talk about biology. Because to me this felt pretty good, but I want to make sure that that's reflective of my audience here. Yeah. I mean, because I felt like we were vibing today in chat. I feel like y'all were very engaged with what I had to say, which is so exciting. Because sometimes it can be quite hard to even get my students to give me answers to questions that I ask, and they're trained in biology. So it's nice to have an audience that is, well, for some of you who want to participate, that you are eager to participate. And since you don't have an incentive to suck up to me, that's like, it's like a thing. Testing 1 2 3. Okay, we're back. Don't know why that happens sometimes. but yeah, so I don't know how much cut out, but this is very good in terms of the spread that I think is relevant, right? Like the way that I would deliver this to biology professionals would be kind of different. So I'm happy to see so many people interested in biology who maybe are not professionally interested the way that an undergrad biologist would be. But I'm also glad that undergraduate biologists are here too. Hello, hello. You're probably nearing the end of your school year, and the fact that you're seeking out more content, I'm rooting for you. Biology is such an amazing discipline. For the virtue of it being interesting. I also wish you good luck in your careers in the future. And to all of those of you who are enjoying biology sort of casually, I'm very excited to bring you some of the best and most exciting stuff that biology has to offer. Yeah, it's pretty incredible. Okay. Yeah, should I chat? Should I go? I'm kind of hungry. If I were to chat, what would I chat about? Imposter syndrome. We started the stream with that. Now there are more people in here. I'm wondering if that's something to talk about. Also, I'm Moroccan. Welcome. Very cool to have viewers from all over the world, not just in my neck of the woods. So here's a question for those of you who are biology undergrads. What are y'all interested in doing? And if you're in high school, what are y'all interested in doing? I guess anybody can tell me what they're interested in doing. If you're in a position where your career might be changing, I'm just kind of interested, because as an instructor, part of my job is to ensure that the egos of my students are well preserved. It's kind of interesting because I've said this on stream before, but my colleagues who are teachers at like high schools and middle schools and stuff, I always praise them with the fact that not only do they have to be teachers, but they also have to be therapists. And as an instructor at the university level, I don't have to do that as much, but instead of being a therapist, I kind of have to be like a career counselor instead. So I'm kind of interested in what chat's thinking about. It's also near the end of the school year for certain cycles, so that's also something interesting to reflect on. University is cheaper in Europe. Yeah. A lot of European countries take better care of students than we do. Been interested in trying to advance medicine. Had interest in making cool pets, plants. You might hate my DIY opinion. So if you want to get into DIY biology, I highly recommend you get professionally trained. That's a big deal to me personally, and that could be taking a lab class at a community college. So even if you like, I do not endorse practicing biology because there's just so much that we don't know. Part of what I hope to impress upon you all is a sense of scientific humility in the way that I talk about science myself. Like, I am not the ultimate arbiter of biology. I don't know everything about biology. There's a lot that I don't know, and I know that I don't know a lot. And so it's always best to practice extreme caution when it comes to working with stuff that you're not familiar with. Though citizen science, like helping... I don't remember. There's a citizen science project that allows you to fold protein and to help scientists understand how proteins fold. That could be a good entry point for citizen science. A lot of jobs creating collating knowledge is my purpose of life. That's awesome. Yeah, I'm what I would call information addicted. Looking to do neuroscience research. I did neuroscience research. I love neuroscience. Neuroscience is awesome. Good luck to you there. Always want to learn biology, never got the chance. Welcome. I hope you stick around. It's all I ever do. Interested in law as a career, but also love STEM, especially biology. We've had other chatters also doing law, so that's very interesting. You could do an overlap of law and science in terms of what is that called? I don't actually know if lawyers are... No, that's policymakers, because with AI and biology and CRISPR, there's a lot of legal gray zones that I think would be interesting to look at in the future. An undergraduate doing a computer science major and maybe going into a field that involves biology. Computational biology is awesome. yeah, computational genetics, computational biology, big data. I don't have the brain for any of that, but I wish you good luck in that. It's super interesting. Um, I actually dropped my comp bio class because it was too hard for me. So you're already better than me in that field. Emerging my lab classes on computer science. Yeah. Computer science and biology have huge overlap. Don't want to admit that I'm a dropout, but still interested in learning in bio. You can always return to education. There's no shame in admitting that you need a break. In fact, I wish some of my students at some points in my careers would take that kind of break. If school isn't working for you, it just isn't no reason to continue. But I applaud you for the bravery for admitting that. Would it work out to contact professors to get their opinion and have them review stuff before actually working on a project? So the reality is that professors are incredibly busy people. You're probably not going to get a response unless they're, for whatever reason, not busy. Again, I would recommend getting actually trained if you do want to do DIY biology, because, we don't ever want to read a headline where an untrained biologist releases some organism into the wild and then an ecosystem is ruined. Or then, there's a lot of rules to follow, a lot of equipment that you need. But if you want to do DOA biology, fermentation is an excellent safe home experiment that you can do. Like making your own beer, making your own wine, making your own bread, maybe perfecting a strain of yeast that makes you the best bread. I think you can't go wrong with that kind of thing. I worked at a genetics company making electronics for these machines last year, so I got a basic introduction to biochemistry. That's really interesting. I don't have the brain for machinery, and so I applaud you for that level of technical knowledge. Got an internship at the hospital. It was so boring. Yeah, same. That's partly why I didn't do medicine, because the life of someone who lives in who works at a hospital, not only can it ping pong between boring and way too stressful, but it's also too much responsibility for me. Law plus science equals forensics. Oh yeah. True. That's what that would be. Diagnostics, basically working in a lab to help doctors diagnose patients. Molecular biology is the second degree I chose to pursue to make me more competitive once I enter the job market. Yeah, I thought about diagnostics too, in case I ever get fired from my job. I think that would be a lot of fun. I don't think I'd get fired from my job, but you never know. Came back from a 3-year break from school, and the time off definitely helped what I needed before coming back. Yeah, the worst situation is like when you just watch a student just tank their grade, and you reach out and you try to make like accommodations, and then they they just kind of trot on. I think taking action, even if that action is taking time off, is really, really valuable. Yeah. Yeah, I don't think there's any shame in that kind of thing. And I think what's important, is that you remain curious and are hungry for improvement. I don't want to do like guru stuff on this channel, right? I'm not here to give advice. I'm not even qualified to give advice. If you want to be a successful scientist, I'm not a successful scientist by definition, right? I did do science before, but I didn't make my whole career being a professional scientist and then return to teach. My primary passion is teaching. And so what I am qualified is to teach you how to think like a biologist, but how to be a biologist is a different question entirely. Or how to be a good student. I was a great student. I wasn't a perfect student. I had issues as a student, but I wasn't smart enough to not have any issues, which I think makes me a better teacher, right? I grew up with some real smarties in high school that would make me feel so bad about myself because they would effortlessly ace everything that they came across. It felt like they never had to try, but I like busted my ass. I felt like Rock Lee a lot of the time, right? Just like grinding constantly. But there were definitely hiccups along that way, and so I think I've learned a lot from those kind of hiccups. Yeah, tuberculosis and rabies both have historical connections to vampirism and the myths surrounding vampirism, which I thought about like really going in. So I've worked on the rabies video. The peek behind the scenes, the rabies video had a couple of different drafts. I've actually made 2 different rabies videos before this rabies video came out, and I threw away both of them because I didn't like either one of them. And one of them went into depth in terms of the historical context of rabies. But I figured that in this kind of format, it might be better to cut that kind of thing out, because what I want to sell is not the context surrounding pathogens, but I want you to all fall in love with the molecular biology surrounding pathogens. It's kind of my foremost goal. Yeah. Okay. Does anybody know if like gifting membership is a thing on YouTube? Like I read somewhere that you can gift free membership like once every month, and I don't know how to do that. Does anybody know how to do that? I saw a fellow streamer, Faye, gift membership as like the streamer, but I don't know how to do that. I should have looked at it before the stream. I totally forgot. What do you think about doing Philoviridae? So I'm guessing this is Ebola. So I looked into Ebola just very briefly, very brief look into Ebola, and the immune-evasion stuff wasn't particularly interesting. The mechanisms by which it inflicts bodily harm are probably interesting, but I think are off-brand for me in particular, right? Because, again, I don't want to do like gore or human misery. I want to do molecules and cells and stuff. Would you make a video on the history of molecular biology? That would be fascinating. So I thought about doing a Darwin series, and I actually have a full script written about the man Charles Darwin, because Charles Darwin is, I think it's fair to say, the closest thing I have to a scientific hero of mine. Now, Charles Darwin as a human was not a perfect human, of course. 1800s, a lot of problematic beliefs about the world, but in terms of, he wrote The Origin of Species in a way that it didn't piss off the entire religious world, was crazy to me. He's such a good writer. And as someone who's very invested in communication of science, Charles Darwin is one of the goats of science communication. Let's see. I don't know if I can do it for free on my end. It doesn't seem like I can. Dang. I could have sworn that I heard that you could do it for free, but maybe not. Darwin's regrettable opinion on barnacles. I actually don't know what you're referring to. What I'm referring to are some like kooky sexism and racism, but you know, who wasn't back then, right? I wanted to do a video where I covered his love letters between him and his wife. Also problematic, he married his cousin, so that's not great. But he wrote so well. He had so much riz, and he was so cute in those letters, and he was so expressive and communicative that you really got a sense that Darwin was actually a very moe kind of person, and that level of humanity I think is really interesting. I wanted to transition into covering The Origin of Species, just because The Origin of Species is kind of the founding document of biology as a discipline. So before Origin of Species, biologists were called naturalists, right? There wasn't really a concept of cell molecular biology, but after The Origin of Species and after developments with the microscope, you got a lot more of what you recognize today as molecular biology. The crazy thing about the history of molecular biology is that it happened so fast, right? It hasn't even been like 100 years since molecular biology got the ball rolling, right? Like, I think that's incredible that we've went from like not even knowing what DNA did to being able to write any DNA sequence we want in a matter of less than 100 years. It's just absolutely crazy. Okay. All right. I think I'm probably going to call it there because I am getting pretty hungry. But I'm very happy that you all came out to hear me talk about rabies and then ramble about science. I hope you all have a good rest of your day. For those of you who are studying for finals at whatever education level you're at, I wish you good luck. And I hope to see you in the next one. Keep an eye out, by the way, for, you know, things that might be going on my channel, specifically on May 19th. And stick around for the loading screen if you haven't seen that. I worked really hard on that loading screen. I think it's awesome. And yeah, I am going to go ahead and stop the stream. Stick around for the end screen for a little bit. It's very fun. And yeah, goodbye, y'all. Goodbye. Oh, whoa, 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 whoa. Well, okay, now I can't leave. Thank you, Sang, so much for the 10 gifted membership. Holy! That's awesome. Thank you so much. Yeah, incredible. Absolute goat. Absolute legend. Dang. So, for full transparency, I live quite comfortably. I don't need this to survive. But all of this money that comes through membership goes to supporting Toxo, who does all of the art for my channel. She's very reluctant to have me, like, she does it free, right? She doesn't like take payment from her partner, which is me. But I made an agreement with her that if I made money through membership, that I would treat her to omakase. And I think we are pretty close, actually, to having a fully funded omakase fund, at least if we ever go to Japan, right? So in the US, omakase is stupid expensive. But if we have a Japan trip, you've paid omakase for Toxo, which I'm incredibly grateful for, because it's so hard to show my appreciation through anything other than words, and I just feel like that's not enough sometimes. Thank you so much, absolute goat. Thank you. That's incredible. All right. With that in mind, bye, y'all. We're gonna head to the loading screen. Bye.